So we just learned about how traits are inherited following Mendel's laws and Mendel's rules with complete dominance. But now we're going to look at other real world situations where things don't often fit into Mendel's perfect little package. So the main thing that we're going to look at first is dominance. Mendel had this idea of complete dominance, where one allele is explicitly dominant over the other. The capital allele will always overpower the lowercase allele. But that's not true in the real world. You'll have two particular instances where this does not follow the idea of complete dominance. The first one is incomplete dominance. This is where neither allele is completely dominant over the other. Essentially, they're sharing dominance, or they're both trying to be dominant, but they fail at it. So in this case, if I have the F1 generation, it's going to create a heterozygote, and that heterozygote is going to be a blend or a mix of the two parents. So the example here is a red flower and a white flower creating a heterozygote offspring that is pink, which is a blend of the phenotype between the two. You'll then see really fascinating ratios when you let the F1 generation self-fertilize. You'll end up with 25% showing one of the different alleles, and then 50% showing that blended allele. So this is common in flowers like snapdragons or carnations. You'll also see it in fur color of some animals like bunnies or dogs, things like that. Now, codominance is where both alleles are dominant. Neither one wants to budge, essentially, in showing its trait. So when you have a heterozygote, you're actually going to see both traits manifested at the same time in the phenotype. The main example with this one is with cattle. You can have a red cow with a white bull or vice versa, and that will produce a red and white speckled offspring that we call roan, like in this picture here of the cute little cow. These ones, similar to the incomplete dominance, are written a little bit differently when we're writing their genotypes. You can either pick a specific gene, for example, the one that I'm giving you the example of here, the C stands for like fur color or for flower color, and then you will add a superscript to explain what allele you're looking at. So you can have the red superscript, the white superscript, and when you have the case of a heterozygote where you have an R and a W, that's when you see the roan phenotype showing through. Another example you can see in this flower here, the flower is showing both white and pink or red characteristics at the same time. An example that hits very close to home is the human ABO blood typing. You have type A blood, which shows one type of antigen on its surface, and type B blood, which shows a different antigen on the surface. These these come back to surface carbohydrates. Each of these has a different particular carbohydrate that they're expressing on their cell surface. But in type AB blood, I am going to have both of those carbohydrates expressed on the surface of my my blood, excuse me. So I will have both A and B characteristics in one blood cell. So that's a codominant situation. Blood also takes us into a different type of inheritance pattern, and that is there's not always just a dominant and a recessive allele. You can have multiple alleles for one particular trait. So in this case, we have an allele for type A blood, an allele for type B blood, and those two are codominant. And then we have an allele for O blood, and this one is actually recessive. So you have a couple different inheritance patterns that are going to show up in just your ABO blood type. You're going to have the possibility of a homozygous or a heterozygous person when you're looking at type A and type B bloods. So if we look at the example down here, I can have a homozygous individual that has 2 alleles for the A particular type of blood, or that person can be heterozygous. They can have 1 allele for the A and the recessive allele for O. Only when we have 2 recessive alleles will we see type O blood. And type O blood is just characterized by not having any of those surface carbohydrates or not having the same chains as AB or AB blood. Another way that we can inherit our characteristics is through pleiotropy. And this is where one gene affects more than one phenotype. And if you really stop to think about it, this makes sense. And it would make even more sense that it's happening quite frequently in our body. Most of your genes are actually pleiotropic. And this means you're going to create one particular protein, and that protein is going to be used in many different areas of your body. I can have a protein that makes up my cell membrane channel proteins, and that's going to be expressed in all of my cells or in most of my cells. So you're going to have one gene affecting many different parts of my body at the same time. An example of this from Mendel's experiments is pea color. So the pea flower itself, that particular gene is going to be linked to the type of outer coating of the seed. So you have 2 genes that are going to be affected here. And then an example in humans of this producing a different phenotype is achondroplasia. It's a form of dwarfism where you have one gene being affected, but it's going to then carry over into your height, your limb formation, and all of these other characteristics. Then there's epistasis. And this is where the expression of one gene is going to affect the expression of a different gene at a completely different locus, at a completely different location. One gene is going to hide or mask the other one, almost as if we were talking about dominant and recessive genes. Now, it's not really how it works, but that's a good way to think about it to keep the idea straight in your mind. The example that your book uses, and that is really easy to understand, is this idea of fur color in Labrador retrievers. So the fur color for these labs is going to be controlled by a B gene and an E gene. The B one determines whether or not fur is going to be black or brown. The capital allele is going to be linked to the black fur coat, and the lowercase allele is going to be linked to the brown or the chocolate fur coat. So in order to get a chocolate lab, you would have to have 2 little alleles. To have the dark black coat, you can have either the homozygous dominant or the heterozygous situation. But we have to take into account the second gene, that E gene, and this is going to control how that fur color, that protein that creates the color, is deposited into the fur cells itself. So if we have a capital E, then the fur is going to be, the color is going to be distributed into the fur the way we would normally expect. However, when you have 2 recessive alleles, when you are homozygous recessive, you're going to end up with no color penetrating into the fur itself. And you're going to end up with a cute little yellow Labrador puppy. So this yellow lab may have 2 dominant alleles to make it have black fur color, but because it is homozygous recessive for the E gene is hiding the expression of the B gene. So no matter what alleles it has for B, a puppy with little e is going to show up as a yellow lab. You also have traits that are polygenic. Poly meaning many, and genic meaning genes. So these are traits controlled by many different genes. And what will happen is the phenotype of these particular traits are going to seem to have a blend, or kind of like a continuum of colors, similar to what you see down here with skin pigmentation. There's not just 3 different sets of skin color, light, a medium, and a dark. There's a gradient or a shading that occurs in between all of it. You can have very, very dark or somewhat dark, or darker brown, lighter brown, very, very pale, or a little bit more olive. We have a lot of different options there. The same thing with like our eye color. You can have very, very dark brown eyes or lighter brown eyes, or you can even have honey colored eyes or hazel. So these particular genes and traits where we seem to have a continuum or a gradient of possibilities are linked to many genes controlling that one trait. Skin color, for example, is thought to have almost 400 genes controlling what the final phenotype will look like. So we have 23 pairs of chromosomes. 22 of these pairs are considered autosomal chromosomes. These are the ones that control normal daily functioning of your body. But then we have that 23rd pair, and these ones are going to primarily be there to determine your sex. We call them your sex chromosomes because they're going to determine whether you are genetically male or genetically female. And these sex chromosomes, you can have X's or Y's. We have 2 different versions of these chromosomes. Females inherit 2 X chromosomes, males inherit 1 X and 1 Y chromosome. That Y chromosome is really small, and it really only has 1 gene of importance, and that is the SRY gene. And that is the gene that essentially tells the body to create the male phenotype. In absence of that gene, the body will develop as female. The Y chromosome doesn't really have many other traits associated with it aside from that SRY one. So all of the traits on the Y chromosome are going to be related to maleness. Now, these sex chromosomes were discovered in the early 1900s by T.H. Morgan. He was kind of one of the pioneers of genetic research in the modern age. And when he was looking at mutations, he found that when he got mutated colors of eyes in his fruit flies, they were almost always linked to the sex of the fly as well. Only the males seemed to have the mutated eye color. So this led him to believe that there was a particular chromosome or a factor of inheritance that was going to be related to the sex of the organism. Now, these X-linked traits, we have 60 or more that we found are related to traits that are not related to whether you are female. So the X, while it is a sex chromosome, has information on it that is important to everyday life and affects traits that aren't related to maleness or femaleness. So for example, certain traits that are linked to the X are colorblindness, that red-green colorblindness, or hemophilia, the ability of your body to clot its blood properly, or Duchenne muscular dystrophy. You can see in the graphic here all of most of the particular traits that are associated on that X chromosome. Now, the way we write these particular traits is similar to how we did with the incomplete and codominant traits. We'll have the X chromosome and then a superscript describing the gene itself. You'll have capital letters for dominant, lowercase letters for recessive. So in these cases, females can inherit 3 different possibilities. They can be homozygous dominant, heterozygous, or homozygous recessive, because females inherit 2 X chromosomes. Males, on the other hand, only inherit 1 X chromosome, so they only have 2 options. They can be homozygous, or they can be dominant or recessive. They won't have the possibility of being a carrier like the heterozygote female. So females that have one dominant and 1 recessive allele are carriers, and they can pass it on to their children. Males either have the trait or they don't. So when we look at pedigrees linked to these X-associated traits, we have a couple different things that we're looking out for, and it will determine whether they are recessive or dominant. Now, with recessive X-linked, you're going to see more males affected than females, and that's because the males either get the good copy or they get the bad copy. There's nothing in between. So if you look at the pedigree at the top right here, you can see that I have 1, 2, 3, 4, 5 males that are being affected, but only 3 females. Normally, you may even see more dramatic differences in the sex layout with these pedigrees. You may see 10 males and only 2 females. It's definitely a trait where you're going to see the males more affected than the females. And this is an example of something like hemophilia or that color blindness. These are recessive disorders. Then you have the dominant disorders. And for these ones, you're going to have a pretty much equal number of males and females affected, because now we're looking at the capital letters, so females can be affected as well. So in this case, we have to look at who is passing it on. Males can give an X chromosome to their daughters only. They give their Y chromosome to their sons. So if they have an affected X chromosome, they are going to give that X chromosome to all of their daughters. So any male that's affected is going to have all of their daughters affected. Females can give it to either their sons or their daughters, because females have 2 X's, so they are going to give 1 X to their sons, and they can give either one of their X's to their daughters. So it would just depend on whether or not the female is homozygous dominant or heterozygous. If that female is heterozygous, there's the possibility that they can give an unaffected allele to a daughter, and their daughter would be unaffected, or they can give that affected allele to a daughter, and the daughter would have the disorder. So you just have to pay close attention when you're looking at these particular types of pedigrees. Now, a really interesting one, we've actually been able to trace the cases of hemophilia in the European royal family. And it all seemed to start with Queen Victoria of England here. And you can see that we've got many males affected, but no females actually had the disorder. All of the females were carriers. So it was hiding in the royal family, but because the royals would only marry other royals, we saw this trait pop up a lot more. You had a lot more heterozygous individuals crossing, creating homozygous recessive offspring. Now, with this particular one, there's no history of hemophilia in the royal family prior to Queen Victoria. So what had to happen there in order for this disease to suddenly manifest? Well, the thought is that she experienced a mutation in her genome that caused her to display this hemophilia disorder on her X chromosome. So she had an X chromosome with the mutation that then caused her to pass it on to the rest of her family. Now, females, as I said, inherit 2 X chromosomes, but you only need 1 to live. In fact, living with 2 often causes some other issues in the body. So females have to deactivate one of their X chromosomes, and this occurs when you're in embryonic development in utero. So whatever X you inactivate is inactivated permanently. The inactivated X is called a bar body because it's not really going to be helping us anymore. So it's not really a chromosome, it's an inactivated chromosome, so it gets its own special name. Now, in animals, we get a really cool pattern of inheritance because of this X inactivation. It doesn't always work that one X is always deactivated in every single cell. You'll often have a patchwork instance where the X is going to be deactivated differently in every single embryonic cell. So maybe it will deactivate the X that has the allele for orange fur color in one cell, but in the other one, it deactivates the one that corresponds to the black color of the fur. So you end up with an embryo that has a different color fur being expressed in different cells. And when you end up with the organism being born, it shows up as these really cool fur colors in cats called calico and tortoiseshell. So they will have orange fur being expressed in some parts of their body because the orange allele is the active one and the black allele was deactivated. And then the black allele is going to show up in other places because it was the one that was kept normal and they deactivated the orange color. So you get these really awesome patterns all because of that X inactivation. So this means that calicos and tortoiseshell cats are only females. If you ever have somebody saying, oh, I have a tortoiseshell male, most likely that's not true. It's very, very rare to have these because you're going to be looking at a male who has another chromosomal disorder. They're going to inherit more than one X. So you can have males with it, but it's far, far less common. And the last thing we're going to talk about are these mitochondrial disorders. Our mitochondria have their own DNA. Think back to that idea of the endosymbiotic theory, where our cells engulfed early prokaryotes and we learned to live in symbiosis together. So these particular organelles have their own DNA still, and their DNA can be mutated just like ours can. So there's certain types of mitochondrial mutations that lead to disorders or diseases in the organism itself, in the person. Our mitochondria are only passed down through the egg. They're not passed down through sperm. So you get all of your mitochondria from your mother. So that means when we're looking at these particular mitochondrial disorders on a pedigree, the males are not going to be able to pass this on. So if you have an affected male, none of their offspring will be affected because the male's mitochondria never make it to the offspring. You will only see a pattern of inheritance between the females. A mother will give it to all of her children, but the father will never give it to any of his children. And that's all I have for you today.